Hi, I'm Dr. Felipe-Andrés Piedra, an instructor at Baylor College of Medicine and research scientist with the Texas Medical Center Genomic Center for Infectious Diseases. Today, I'm going to talk to you about improving our understanding of variable RSV disease severity through measurements of viral dynamics and host response in multiple in vitro infections. Most of the data I'll present come from a manuscript that I co-authored, that I co-first authored, and that we recently uploaded by our archive and submitted to the Journal of Virology. So RSV stands for respiratory syncytial virus, which is also known as human RSV or human orthopneumovirus. Like SARS-CoV-2, RSV is an enveloped RNA virus. Unlike SARS-CoV-2, it belongs to the viral order Mononegavirales, which consists of viruses containing non-segmented negative sense RNA genomes and includes other important human pathogens like Ebola, measles, and rabies. RSV is a pneumovirus containing two major subgroups, A and B, estimated to have diverged over 300 years ago. RSV infects ciliated cells of the respiratory epithelium via what has long been thought to be a process of direct fusion of the viral envelope with host cell, but recent evidence suggests it is also taken up by its host by macropinocytosis before undergoing fusion and depositing viral genetic material into the host cytoplasm. Infection is mostly asymptomatic to mild, but can lead to severe lower respiratory tract infection and even death, especially in young children, the elderly, and the immunocompromised. RSV is therefore an opportunistic killer. Globally, young children are hit hardest, especially preterm infants and neonates. Over recent non-pandemic years, there have been on average over 3 million hospitalizations and over 100,000 deaths per year worldwide in children less than 5 years of age. 99% of these deaths occur in developing countries, a figure that starkly reveals the crippling effects of poverty and societal dysfunction beyond the lack of economic opportunities we tend to emphasize. Indeed, the pandemic we're living through is teaching us much the same lesson. In the US, RSV is associated with over 200,000 hospitalizations per year, mostly in the very young and elderly. Consistent with global data, lower income Americans are at greater risk of hospitalization from severe RSV disease. Known medical risk factors for severe RSV disease include chronic cardiovascular, neuromuscular, and respiratory conditions, rare congenital or genetic conditions, and immunocompromising conditions of all kinds. Known environmental risk factors include persistent exposure to secondhand smoke, other forms of air pollution, and sources of psychosocial stress, from physical and emotional abuse at home to family conflict and community violence. It's important to note that these two sets of risk factors contain elements that are both independent of and deeply related to each other. Regarding the latter, it is increasingly appreciated how chronic stress, for example, psychosocial or respiratory stress, can drive the emergence of disease states that lead to greater susceptibility to severe disease from pathogens like RSV. This brings us to our present study investigating some sources of variable RSV disease severity through in vitro RSV infections. Specifically, our goal was to complete a systematic characterization of RSV infections in vitro using a representative set of subgroup A and B RSV strains. The Ontario and Buenos Aires strains are more similar to circulating strains than GA1 and GB1, which were isolated decades ago, and also using two of the most widely used cell lines in studies of RSV to date. We wondered, to what extent is the host response strain sensitive, and also to what extent is the host response cell line independent, or cell line dependent, excuse me. To answer these questions, we measured viral gene expression patterns by high throughput short read sequencing and viral replication rates using both qPCR and plaque assays. And we measured the host response through gross morphological and cytoskeletal changes induced by RSV, through transcriptional changes or changes to host gene expression, and through changes in levels of secreted cytokines. Now we'll go through our major findings. Let's first look at non-gradient patterns of viral gene expression. This plot shows steady state gene expression levels from all 10 RSV genes and for the 4 strains used. We showed in a previous study that these patterns are cell line independent, which makes sense given that they result directly from interactions among viral proteins of the viral genome. The textbook model predicts a smooth drop in gene expression going from the most 3' gene of the genome, NS1, to the most 5', L. But that's not what we see. An important part of my ongoing work related to my training as a biophysicist is the development of a mechanistic model that captures these data. From the sequencing data, we could also calculate how often a viral polymerase would, instead of terminating after fully transcribing a gene, continue or read through to the next downstream gene. This plot shows rates of read through for the 8 gene junctions or intergenic regions of the RSV genome. Note there are 8 instead of 9 because 2 of RSV's 10 genes have overlapping open reading frames. 2 differences stick out, but closer inspection revealed the NS1-NS2 difference to be likely artifactual. The MSH difference appears real and has never been documented before, and suggests that RSV-B infections lead to less production of SH protein, which is a viroporin. We're exploring the consequences of this difference for the RSV life cycle in other studies. Moving on, we measure the number of plaque-forming units or infectious virions at 4 different time points for infected Hep2 and A549 cells. On average, Hep2 cells showed 10 to 100 times more infectious virus than A549 cells. So clearly, RSV replicates to much higher titers in our Hep2 cells, which is consistent with years of observations made by our lab. Next, we looked at host transcriptional changes for evidence of divergent responses consistent with the differences in viral replication. The data here show the kinetics and magnitude of the global host response to RSV infection. The total length of each bar is proportional to the number of genes analyzed. The length of each red segment shows the proportion of genes undergoing a significant increase in expression relative to mock-infected cells. And the blue segments show the same, but for decreases in expression relative to mock. Focusing on any given quadrant, you can see that the growth through time in blue and red segments is comparable between cell lines. While the size of the host response to RSV infection is cell line independent, the form of it is not. We use the gene set enrichment analysis tool to look for functional pathways enriched for up or down regulated genes. The heat map shown here depicts these data with deep red blocks corresponding to infections and time points showing highly significant upregulation of the relevant genes, and deep blue blocks showing highly significant downregulation. It's clear that the patterns are different between cell lines, but we have to look more closely to determine how. So if we look more closely by plotting z-scores through time, we can see some stark and informative differences between cell lines. For instance, genes upregulated by interferon alpha and gamma are never upregulated in Hep2 cells, while they undergo gradual and ultimately very strong activation in A549 cells. The origins of this difference are not clear, but the difference itself is clear evidence of a more potent antiviral response from A549 cells. If we look at genes encoding inflammasome components, we see somewhat erratic dynamics, but with strong upregulation in Hep2 cells, while A549 cells show weak downregulation and no change in regulation at all. These data are consistent with a more pro-inflammatory response from Hep2 cells. Finally, we can gain more insight into the global character of the response of these cell lines to RSV infection by looking at patterns in the levels of cytokines showing sharp increases relative to mock-infected cells. Infected Hep2 cells show large and comparable increases in the pro-inflammatory cytokines IL-6 and IL-8, while in 3 of 4 infections, A549 cells show much smaller increases in IL-8. Moreover, A549 cells show large increases in RANTES or CCL5, a chemoattractant, while Hep2 cells do not. In general, A549 cells show greater increases in a greater diversity of cytokines, which is consistent with a transcriptional response that appears more potently antiviral than the more pro-inflammatory response of Hep2 cells. Moving on, although most differences observed were cell line dependent, we found some evidence for strain dependence in the host response, especially in A549 cells. I'll show 2 examples. In the first, we observed a sharp downregulation of genes involved in protein secretion in both cell lines and in response to infections of both contemporaneous strains used. We don't have enough time to go into this, but we speculate this might result from a shared duplication in the gene encoding RSV's heavily glycosylated and multifunctional attachment protein. The second example shows genes involved in the TGF-beta signaling pathway undergoing subgroup-dependent regulatory dynamics in A549 cells. To finish, I'll show intriguing evidence of host response features, again appearing more strongly in A549 cells, in apparent support of the RSV life cycle. For all 4 infections, A549 cells show strong upregulation of genes involved in cholesterol biosynthesis, fatty acid metabolism, and not shown here, adipogenesis. These changes might result in the increased production of cholesterol-containing lipid rafts, facilitating both viral entry and egress. In summary, our observations of RSV gene expression suggest the current textbook model of mononegovirales gene expression needs to be revised. We also discovered an unexpected difference between RSV subgroups and gene expression that we're investigating now. On the host side, and in combination with rates of viral replication, the A549 response we observed on the level of gene expression changes and increased cytokine levels was more antiviral than the more pro-inflammatory response of Hep2 cells. A549 cells showed greater but still slight sensitivity to the kind of RSV strain used to infect, and aspects of the A549 response appear to support the RSV life cycle. In total, the A549 cell response indicates it is a more natural host of RSV infection, which is consistent with its origins and evidence of helicontamination in Hep2 cells. Finally, our work suggests that we can use these cell lines to model and characterize the effects of certain environmental factors associated with more severe RSV disease. For example, we might expect a certain pollutant that we can include in our growth media to shift the A549 cell response to the more pro-inflammatory response of HEP2 cells in a dose-dependent way. We can also see a viral replication increases along with it. So thank you for giving me the chance to present some of my work, and I want to give thanks to all my colleagues and friends at Baylor College of Medicine and the TMC Genomic Center for Infectious Disease.